I am Dr. Khokon Kumar Dotto. My Christian name is Dr. David Moon. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Mitochondrial thioredoxin interacting protein Txnip is the main culprit behind the fatal consequences of myocardial infarction or heart attack. Khokon Kumar Dutta et al. For the first time discovered the presence of thioredoxin-binding protein 2 TBP-2 inside the mitochondria of rat renal proximal tubular cells in 2005. We also confirmed the cytoplasmic and nuclear locations for TBP-2 for the first time using direct assay method. The report was published in the pathology section of Nature Laboratory Investigation. TBP-2 is known as thioredoxin interacting protein Txnip in humans. It is also known as vitamin D3 upregulated protein 1 VDUP1 as it is the most sensitive and responsive gene under vitamin D3 application or treatment or administration in the human body. We also reported the physical interaction between mitochondrial thyroidoxin 2, TRX2, and TBP2 in vitro. This was also the very first time discovery internationally. TRX2 in normal condition neutralizes free radicals inside the cellular mitochondria. TBP2 inhibits antioxidant activities of TRX2 by blocking the thiol groups in the active site. It is to mention here that there are many sites inside the cells that generate free radicals, including the mitochondria. But the major part of free radicals are generated inside the mitochondria, which is mostly very toxic and harmful for humans and other species on this planet. Mitochondrial respiratory chain is a good source for free radical production. It is suspected that the components of thiol defense system and the respiratory chain may have some sort of crosstalks and involvements in the ROS production inside the mitochondria. The detailed functions and importance of mitochondrial TBP2 or Txnip or VDUP1 remained unknown for several years. After its first time discovery that was published in Scientific Journal in 2005. At present, it seems that mitochondrial Txnip or TBP2 or VDUP1 or translocation of Txnip or TBP2 or VDUP1 inside the mitochondria is one of the most critical events for good health and diseases in humans at least. Here I would like to focus on a recently published research article where mitochondrial activity of Txnip or TBP2 or VDUP1 was reported as the main culprit behind the dangerous consequences of myocardial infarction or heart attack, which in many cases lead to heart failure and eventual death. Yoshinobu Nakayama et al. Published an article online in February 2021 in the Journal of Molecular and Cellular Cardiology entitled Txnip C247S mutation protects the heart against acute myocardial infarction. The article will actually be published in June 1st, 2021. Txnip C247S is a mutant form of the wild type Txnip. The cysteine residue at 247 position of Txnip is important to make disulfide bridge with thioredoxin and to block the ROS scavenging function of thioredoxin. When cysteine is changed with serine at 247 position, Txnip cannot build up disulfide bridge with thioredoxin and cannot stop antioxidant activity of thioredoxin. The group firstly confirmed the overexpression of Txnip C247S results in reduced level of ROS inside the cells. In hypoxic condition, high presence of Txnip C247S in the cells reduced the killing effects of wild-type Txnip in compared to the control cells. Low level of ROS was found in the presence of high Txnip C247S under hypoxic condition in compared to the cells expressing only wild-type Txnip under hypoxic condition. To note here that blockage in the artery often reduce the blood flow in the heart or specifically in certain part of the heart and creates a hypoxic condition that initiates the deleterious effects of heart attack. The group also checked the effects in animal model. Cardiomyocytes specific Txnip C247S knock in mice had better survival with smaller infarct size following myocardial infarction compared to control animals. The absence of thioredoxin inhibiting activity of Txnip promoted better antioxidative capacities in mitochondria of cardiomyocytes that protected from oxidative damage induced by myocardial infarction. An unbiased RNA sequencing screen made by the group revealed that hypoxia-inducible factor 1 signaling pathway was involved in Txnip C247S mediated cardioprotective mechanisms. At present, cardiovascular disease is the number one killer of humans worldwide. Some of the cardiovascular disease raise myocardial infarction or heart attack on their way to creating fatal consequences. Hope the world would get benefited to a large extent targeting Txnip or inflammation signaling or inflammation in the fight against cardiovascular disease, especially heart attack and heart failure. Finally, I would like to wish good health and better days for you everybody. Thank you.